Embryoid bodies are aggregates of embryonic stem cells. The most common way of creating these aggregates is the hanging drop method, a laborious approach of pipetting an arbitrary number of cells into well plates. The process is manually intensive because the media in each well plate has to be manually exchanged each day. Moreover, though recent studies have shown that the initial number of cells forming the aggregate can have effects on stem cell differentiation, there is no way of predetermining the number of cells using this method. In this video, we show a way to simply and rapidly load different numbers of cells into microfabricated wells for embryoid body development. Hi, I'm Jonathan Pegan from the lab of Khine in the School of Engineering at the University of California, Merced.
And I'm Jesus Luna, also from the Khine's lab.
Today we will show you a procedure for creating embryoid bodies in microfabricated wells by centrifuge.
This procedure involves the following steps: creating a micro device, loading the stem cells, and feeding the stem cells.
So let's get started. The first step in this procedure is to make the Shrinky-Dink mold. Using a good definition printer, print the desired pattern on Shrinky-Dink sheets. Next, bake the Shrinky-Dink sheet at 160 degrees Celsius for about 10 minutes, or until it has fully shrunk and acquired a regular shape. After the Shrinky-Dink mold has cooled down for 2 minutes and is cool to the touch, submerge it in an isopropanol bath